Hello, curious chemists. Ever wonder how your body manages to speed up thousands of chemical reactions every second without breaking a sweat? Well, meet enzymes, nature's very own speedy helpers. Here's a little enzyme joke to get us started. Why are enzymes such good friends? Because they always have your reaction rate covered. Enzymes are biological catalysts, which means they speed up biochemical reactions without getting used up themselves, like a chef who cooks amazing meals but never eats a single bite. What makes enzymes truly special is their remarkable specificity. Their active sites, with precise three-dimensional shapes, fit only certain substrates, almost like a lock and key or a personalized handshake. Thanks to this perfect fit, enzymes can boost the speed of reactions by factors as huge as 10 to the power of 17. That's 100 quadrillion times faster than without them. How? They lower the activation energy, which is like making the reaction hill easier to climb by stabilizing what's called the transition state complex, kind of like holding the door open for the substrates to turn into products. Picture the diagram with the enzyme as a glove and the substrate as a hand. The active site snugly fits the substrate, ready to jumpstart the reaction without the enzyme getting changed in the end. This amazing efficiency and precision are why enzymes are essential for life, driving everything from digestion to DNA replication. Welcome back. Let's now explore how enzymes are sorted into category, kind of like organizing superheroes by their powers. Enzymes are grouped into 6 major classes based on the types of reactions they catalyze: oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. Think of oxidoreductases as the electricians handling oxidation and reduction by moving electrons around, while hydrolases are the plumbers breaking bonds by adding water, like chopping long chains into smaller pieces. To keep track of these enzyme superheroes, each gets an enzyme commission or EC number that codes its class, subclass, subclass, and identification number. Imagine a superhero's secret agent number. Their names usually end with SA to indicate enzyme activity, like lactate dehydrogenase, which helps turn lactate into pyruvate in your muscles during exercise. Hello again. Have you ever tried fitting a key into a lock and noticed some don't fit at first but mold into place with a little wiggle? This idea leads us to how enzymes work by forming an enzyme-substrate complex, abbreviated ES, which then turns into product and frees the enzyme for another round. There are 2 main models here. The lock and key model imagines the enzyme's active site and the substrate as rigidly fitting together like a perfect puzzle piece, straightforward and neat. But biology loves flexibility, so the induced fit model adds a twist. The enzyme changes shape slightly when the substrate binds, wrapping around it like a hug for a perfect fit. This adaptability is what makes enzymes so effective in catalysis. Enzymes can speed up reactions by acid-base catalysis, covalent catalysis, or by bringing molecules close together to make interactions easier, all little tricks to get chemistry done faster. The reaction pathway can be written as E plus S reversibly forming ES, which then produces E plus P, meaning enzyme and substrate combine, then yield enzyme and product without the enzyme being used up. This mechanism explains why enzymes are amazing at speeding up reactions while remaining unchanged themselves. Hey everyone! Let's talk about what makes enzymes happy or grumpy, because their activity depends on several factors. Like us, each enzyme has an optimum temperature and pH where it works best. If the temperature rises from cool to just right, enzyme activity increases, since molecules move faster and collide more often. But push the heat too far, and enzymes start to denature. Their shape unfolds like a bad haircut, making them lose functionality. It's like how you can't work your best if you're too hot. Similarly, pH affects enzymes differently depending on their environment. For example, stomach enzymes prefer acidic conditions. When we keep enzyme concentration constant but vary substrate concentration, the reaction rate zooms up first but then plateaus at a saturation point. Think of a restaurant kitchen where too many orders eventually overwhelm the chefs. Here, the Michaelis-Menten constant, denoted Km, tells us the substrate concentration where the enzyme reaches half its maximum velocity, a critical measure of enzyme performance. In the diagram, you'd see graphs showing enzyme activity changing with substrate concentration, pH, and temperature, revealing peaks and drops that tell us just how sensitive enzymes are to their environment. Understanding these factors helps optimize enzyme use in industries and medicine, ensuring they perform at their best wherever they're needed. Welcome back to the heart of enzyme kinetics. The Michaelis-Menten equation beautifully describes how the reaction rate, called V, depends on substrate concentration, written as capital S in brackets. Two key players here are Vmax, which is the maximum rate the enzyme can achieve when fully saturated with substrate, and Km, the Michaelis constant, which indicates how strongly the substrate binds to the enzyme. A lower Km means better affinity. The equation goes like this: V equals Vmax multiplied by substrate concentration divided by Km plus substrate concentration. In other words, as substrate amounts increase, the reaction rate rises but levels off when the enzyme is saturated, forming a hyperbolic curve when graphed. On this plot, you'll see Vmax marked at the plateau and Km at the substrate concentration where V is half of Vmax. So when substrate concentration is much greater than Km, rate approaches Vmax. But when substrate concentration equals Km, the rate is precisely half of Vmax, a very handy benchmark. This equation and its graph help us predict enzyme behavior under different conditions and design effective inhibitors or activators for various applications, from drug design to biotechnology. Good to see you again. Enzymes don't always work alone. Many require extra helpers called cofactors or coenzymes to get the job done. Cofactors are typically metal ions like zinc or iron, while coenzymes are organic molecules such as vitamins, working as essential accessories that enable enzymatic reactions. But watch out, inhibitors are like party crashers that reduce enzyme activity. Competitive inhibitors sneak into the active site, blocking substrates like a fake key jamming a lock, while non-competitive inhibitors bind elsewhere on the enzyme to change its shape and performance, kind of like hitting the pause button remotely. The good news is that reversible inhibitors can be overcome by adding more substrate or removing the inhibitor, but irreversible inhibitors form permanent bonds, disabling the enzyme forever. Imagine superglue messing up the lock so no keys can fit again. Infamous enzyme poisons like cyanide and heavy metals act this way, shutting down vital biochemical reactions and proving how powerful and dangerous enzyme inhibition can be. Picture the diagram with two parts, one showing substrates and competitive inhibitors competing at the active site, and another with non-competitive inhibitors binding elsewhere to distort the enzyme's shape, giving a clear picture of these inhibitory strategies. This knowledge is crucial for developing drugs that can target enzymes in diseases or to detoxify poisonings. Alright, time to put theory into practice with a quick calculation. We are given a Michaelis constant Km of 5 millimolar, a maximum reaction rate Vmax of 100 micromoles per minute, and a substrate concentration S of 5 millimolar. Our task is to find the initial reaction rate V. According to the Michaelis-Menten equation, V equals Vmax times substrate concentration divided by Km plus substrate concentration. Substituting, we get V equals 100 micromoles per minute multiplied by 5 millimolar divided by 5 plus 5 millimolar, so V equals 500 divided by 10, which simplifies to 50 micromoles per minute. Notice this is exactly half of Vmax, just as expected when substrate concentration equals Km. This confirms the formula works perfectly and shows how enzyme activity responds to substrate availability. Envision on the plot where the point at substrate concentration 5 millimolar lies halfway up the curve toward Vmax, clearly marked and highlighted. Calculations like this are the backbone of enzyme kinetics studies, helping researchers determine enzyme efficiency and guide experiments in biotechnology and medicine. Let's wrap up our enzyme adventure. Remember the basic reaction: enzyme plus substrate reversibly forms the enzyme-substrate complex, which then produces enzyme plus product. That's how enzymes speed things up without being consumed. The key Michaelis-Menten equation relates reaction rate V to substrate concentration. V equals Vmax times substrate concentration divided by Km plus substrate concentration, capturing how enzymes respond to different substrate levels. Keep in mind, irreversible inhibitors form strong covalent bonds with active sites, permanently reducing enzyme availability and halting reactions. Finally, don't forget the 6 important enzyme classes: oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases, each playing unique roles in biochemical reactions. Enzymes really are the unsung heroes of life's chemistry, working tirelessly behind the scenes to keep us alive and thriving. That's it for today's concept. Chemistry is fun when you can laugh and learn. See you in the next session.